Yeah. So, I said that today we are going to talk about ocean acidification and fish gut microbiome. Actually, this is the small work which we done in our Center for Ocean Research. We just mimicked a kind of a ocean acidification environment, and we introduced a marine fish, and we just tested how that gut microbiome content changed and what is its impact. The work. Actually, this particular work we have done with a collaboration of the Department of Bioinformatics, because the sequencing data and all, it is analyzed by the Dr. Jamie Christie and the students Ashin as well as other people. And we also got the technical support from NAOT, particularly Dr. Dharani sir. He helped a lot. Apart from that, Mr. R. Vijayaraghavan, who helped a lot in doing the cake path analysis and all. So apart from that, our teamwork, as I said, myself and Bharat Pandey and Tirigana Subhanan Kumar, who are all involved in this work. And this, before entering into the topic in detail, I just want to tell some of the basic of what is climate change and its impact on ocean, and how this ocean acidification will reflect on as a marine threat. Then I just brief about what is gut microbiome, so that I can narrate the experiment what we did. So this kind of introduction will help you to understand the work in the final slide, why this experiment was carried out and what will be the result. So when we are talking about climate change, everybody is talking about climate change, but in marine environment, climate change is the biggest threat. So burning fossil fuels, like any kind of thing that will emit carbon dioxide, that is fossil fuel, deforestation and industrial agriculture release, as well as everything that will release enormous amount of carbon dioxide. This enormous amount of carbon dioxide that will be taken by the ocean. So 25% of the carbon dioxide is taken by the ocean, and it acts like a sink for carbon. Okay, and due to this enormous amount of carbon dioxide, the entire atmospheric ecosystem is having a global warming condition that will release excessive amount of heat. Even this excessive amount of heat, almost 90% of the heat is also absorbed by this ocean. Because of the warmer ocean and the more acidic environment, there will be a less oxygen atmospheric created in the ocean that will cause six major threat in the ocean environment. They are the sea level rise. Because of this phenomena, sea level rise is accelerating. That will lead to flooding of coastal area, and the coastal communities are very much affected, and drowning of wetlands and other habitats are happening. Then we all know the coral bleaching. The coral bleaching is also because of this warmer and ocean acidification environment, and because of the less oxygen environment happening in the ocean. This bleaching, as it is mentioned here, in warm water coral reefs, particularly the marine biodiversity hotspots, could be lost in the planet because of the 2-degree increase in the normal temperature. Okay. Then toxic algal bloom. Because of this excess of heat, as well as the excess of carbon dioxide sink in the ocean, as I said, there will be a large amount of climate change is happening in the atmospheric, and that lead to a large number of harmful algal bloom in the oceanic environment. Some kind of bloom will even create emitting some kind of toxic thing. Some of the algae, they are almost very toxic to all the organisms which are in the marine environment. That will lead to a mass mortality in the marine environment. With habitat loss. Habitat loss, as if oxygen is getting deployed, definitely people, sorry, not people, the organisms which are dwelling in the ocean environment, they need oxygen, and they, because of the suffocation, the entire population may vanish one day, that will lead to a complete decline in the population. With the ocean acidification, this thing we are going to talk about in elaborate manner on the subsequent slide. And fisheries. So because of this, all these threats, even fisheries population is getting declined because most of the local community people who are living in the coastal area, they depend on the fisheries. And even the fisheries population is getting declined because of the less oxygen, more acidic and warmer environment. So, to narrate the basic of an ocean acidification, this illustration will give a clear idea. This particular tree, actually, uh, the entire chart is divided into 5 phases. The first phase, actually, this is first 3 phases, a normal condition where carbon dioxide is taken up by the ocean by physical process. This is the normal thing. As I said, ocean is being a sink for carbon dioxide. Normally, the carbon dioxide will be taken by the ocean. But when the carbon dioxide combines with the water, it forms carbonic acid, which break down into bicarbonate ion and hydrogen ion. And this hydrogen ion combined with the carbonate ion to form more bicarbonate. This natural process has maintained hydrogen ion concentration, that is in other way the pH of the ocean for thousands of years. So when there is a equilibrium of carbon dioxide saturation in water, there will be a equilibrium amount of hydrogen ion in the seawater environment that maintain a pH of 8.2 in all the area of oceanic environment. Uh, Before the industrial revolution. So once the industrial revolution start, there will be excess carbon dioxide emission happen in the atmosphere. So the higher carbon dioxide emission means there will be more hydrogen ion in the ocean. So there will be more hydrogen ion, and the natural supply of carbon ion cannot be keep place. So when there is more hydrogen ion, there cannot be a uh, which combines with the carbon ion that will form a bicarbonate that will be used for the cell animal. But this higher CO2 emission means there will be more hydrogen ion, and that carbon ion cannot be keep pace. So increased hydrogen ion concentration results in lower pH and also less carbonate, which make it difficult for marine life with calcium carbonate structure. This is the problem with ocean acidification. This is not only problem with the ocean acidification because of the excess of hydrogen ion, the pH is reduced in the seawater. So it will reduce from 8.2 to lesser than that, 8.1, 8, and even 7.9. The change of the pH, that may be a biggest problem for all the marine organisms, where they will live only in an optimal pH condition. So when we are talking about ocean acidification, first we have to tell what are the marine threats. Okay, marine environment is facing a large amount of threats, not only ocean acidification. The first threat is now because of the land degradation, even the mangrove and the seagrass environment, it is almost degraded, and it will be occupied by the population or excess population. Almost 30% of the mangrove ecosystem was decreased and destroyed, and 90% of the decline in large predatory fish stock. This is because of the overstocking. We all know we are trawl netting and industrial fisheries are happening in the ocean environment. Almost they are catching all the fishes, and that will lead to a decline in the fish population. Then 10 million fish sharks targeted by human annually. Generally, sharks will be almost targeted for because of the fins. Okay, the illegal hunting of shark almost decline the shark population in the ocean environment. And some of the people, they are also hunting the whales and dolphins, which are all the marine mammals in the environment, and even it may be killed because of the bycatch, illegal bycatch also. These population are getting declined. Apart from that, there will be a bigger problem. We all know that is because of the marine plastic. Almost 8 to 12 million tons of plastics will be dumped into the ocean annually. Then we all know the mass coral bleach, which is happening every year, and almost the Great Barrier Reef, which is the biggest reef area, even that is getting affected by this. So 90% of coral reef population is almost declined because of this. Apart from this, all these threats, ocean is also facing a major threat called ocean acidification and sea level rising because of the excess of carbon dioxide as well as global warming condition. So what this ocean acidification will do here is the ocean acidification, it will, because of the change in the pH of the water, the entire food chain will get collapsed. Almost it have an impact on the primary producer. They are nothing but the phytoplankton community, and it also affect the secondary population, secondary producers. They are the zooplankton. So even the zooplankton, they are depends upon the phytoplankton. When the phytoplankton population is declined, even the zooplankton may get affected. And because of this, the impact on the zooplankton population, the small fishes which depends on this zooplankton also, that will get affected because they won't get a proper food, and the food chain may get collapsed. If there is no small fishes in the area, the large predatory animal which depends to eat the zooplankton as well as the other fishes, they cannot get any food. So the entire fish food chain will get collapsed because of the excess of carbon dioxide in the oceanic environment. And almost, as I said, the entire population will have a communal effect in the ocean environment that will give a healthy population that may be also altered because of this ocean acidification environment. So this is a graph which shows the carbon emission annually in the world. Actually, this graph is from 1990 to 2018. It is not updated, but even in 2018, you can see the metric ton of carbon dioxide emission per year is 2,277 metric ton of carbon dioxide all around the world. So this is because of the transportation, agriculture, in the building, construction industry, and other energy sector and electrical heat. Because of this, this kind of carbon dioxide emission is happening. Though ocean is almost the transportation, mainly shipping industry is also playing major role in this carbon dioxide emission. Apart from that, the other industrial part all playing a major role in the carbon dioxide emission that will directly have an impact on the ocean. And a healthy ocean should have a 8.2 pH, but it is decreased to 8.1. And in future, it may again get deployed because of the excess of carbon dioxide in the atmospheric, and that is saturated in the seawater. So this particular graph, it is released by National Oceanic and Atmospheric Administration of UN. Actually, this time series graph shows taken from 1955 to 2050. So see, from 1955 to 2050, this red color line shows that there is the excess of carbon dioxide release. So the carbon dioxide release is increased day by day, and almost it reaches, see, almost 400 units it reached. So whereas the dark blue lines are the saturation of carbon dioxide. Even the carbon dioxide saturation is also getting increased day by day. The blue line is also in the progressive phase, that is also increasing day by day. But the light blue color, which is the seawater pH, which shows from the pH scale is shown here. So it is declining because of the increase of the carbon dioxide in the atmospheric, and the saturation is increasing. The pH in the ocean is getting decreased day by day, and almost it from 8.2 it is getting decreased to 8.03. And this particular atlas, the global atlas, which is which was made by Coin, Geong 2000 on 2019. They used a global ocean data analysis project for this. And here you can see the blue region is almost having 8.2.25. This is the normal pH. And this particular blue is the decline pH of 8.15. And this yellow is almost 8. In all the temperate region, the pH is almost declined to 8, almost 0.2 percentage of pH is getting declined. So the temperate waters are the water which is having the most rich biodiversity. On all the biodiversity depends upon this oceanic pH. Thus, the atmospheric O2, that means carbon dioxide, that may have a great impact in the ocean, and they create ocean acidification and ocean warming, and it will also lead to ocean deoxygenation. This condition will have an impact on the increased animal respiration and decrease in the photosynthesis. That again, there will be a more carbon dioxide and less oxygen. And this will have a great impact on the molecular level impact. Even molecular level changes happen in species, every specific species. So this will lead to molecular level changes, organism level changes, and community level changes. Because of this big changes, biological change, molecular as well as organism and community changes, the ecosystem goods and service may happen to get impact. As I said, even the fisheries population get declined, even the coral population if completely destroyed, there won't be any population which is dwelling nearby the coral area. So is indirectly or directly have an impact on the human. So ocean acidification is a kind of a multi-stressed environment for the oceanic environment. Apart from that, ocean acidification, apart from this, ocean acidification is also playing a major role in the acid-base balance in the fishes, because the impaired function of the neural network of the nervous system and brain. As it mentioned here, every fish, the brain system depends upon the surrounding environment, and the surrounding environment should have a optimal temperature and optimal pH and optimal other environmental clues for any host or any fish to live in that particular environment. This particular, if the pH is changing, even the brain function is also get affected. How it is affected means the fish will have an problem on the sensory thing, so that they cannot identify the prey and they cannot eat properly. And this prey, they don't know where these prey, the smaller animal, they don't know how to get their smaller animal, and that population also get declined. Because of this, as they have less sense to feed the prey and food web, the loss of the nutritional balance and growth is reduced. So the all animal, there will be a retarded growth, and there will be any kind of activity in the fishes. Because of the non-activity, there won't be any active reproductive behavior, and there won't be any growth in the population. So ocean acidification is also having a great impact on the acid-base balance of the fish, and this leads to a decline in a particular population, and indirectly decline the dependent population in the eco food system or any kind of food chain. That is the basics of climate change, particularly about ocean acidification, and what ocean acidification is doing to any kind of local community to or other core community. Now I'm just giving some brief about a microbiome. We all made up of genes that we know, but apart from the genes, now there is a new science called microbiome that says that apart from our normal human cell, we are all occupied by the microbes, the bacteria, which are playing the dominant role and that have the tendency to control all the gene function. So as the gene, how genome is, our entire genome is controlling us, like that the entire microbiome, which is native to our body, that is controlling our entire even the cellular process as well as gene expression. Almost 40 million human cells are there, and the total genes are 23,000. But for the same time, it is discovered for human, it is 40 million microbial cells also there, and they are responsible for 3 million genes for some kind of genetical expression. These microbiome, they will be inherited by the mother as well as from the environment and other source, whereas in the case of gene, it is inherited by the parents. They are highly variable, and here these genes are very stable. And if there is an alteration, they will create a disease. Here, the mutation will create disease, and there will be a highly microbial diversity that can be easily monitored. Here, this kind of gene diversity cannot be, it is very limited, and monitoring this is a tedious task. And there is a high degree of plasticity and adaptability. Even though there is a change, it can be easily adapted in the host environment. But gene modulation, that will take to some kind of risk. Here we can even modulate the gene microbiome, and we can get a healthy host. So this is the basic of a microbiome. Microbiome is nothing but a group of bacteria which is living inside the body, maybe or a human body, the whole human body. Yeah, that may be also called as microbiome. If in the fish, even there are a lot of microbiome in the fish body, that is all called as fish microbiome. If it is living in the digestive tract, that microbes or that microbiome is called as fish gut microbiome. So this gut microbiome is not only this particular topic is not only for fishes. It is almost common for all animals. But here we are mainly focusing on fish gut microbiome. This gut microbiome may change, depends upon the age of the fish and the diet of the fish, what the fish is going to eat, and the place where the fish is living. And the malnutrition, if it is not having any kind of proper diet, that is also have an impact on the fish gut microbiome. And if it is cultured in a particular environment, if there is an addition of antibiotic, that may also alter the gut microbiome. And if the cultured condition they add probiotic, that may also alter the gut microbiome. Because of all these factors, this gut microbiome, it may be enriched or it may be get decreased, and it may have a direct impact on the fish genome that will have a healthy growth, or it may lead to a disease. Okay, this interaction is called as genetic variation, and it depends upon the gut microbiome role. The genome or the healthy fish may react to a disease condition or a healthy condition. The healthy condition depends upon the factors like growth of the fish, nutrition of the fish, tolerance to stress. As I said, in the ocean acidification environment, the pH, temperature, everything is changing, and the fish have the tendency to tolerate this particular stress condition. Or if there may be overpopulation also, that have to be tolerated. Then immune system. The immune system of the fish have to be also be expressed in a good manner if when there is a healthy microbiome population is playing a major role in this. And even it will lead to a disease condition in fish if the gut microbiome is negatively doing some kind of role in the fish physiology. So gut microbiome plays a major role in the fishes. So this particular picture shows some of the fishes like eel, salmon, goby, as well as mullet. But all the fishes, they have a digestive tract, and it starts from mouth to anus. And these are all the common parts of the digestive tract: mouth, esophagus, stomach, pylorica, midgut, hindgut, gallbladder, gizzard, and all. So all these region of the digestive tract is occupied by this gut microbiota. As I said, the microbes which are living inside the tract. So this microbiota, they are responsible for the release of the such metabolites called indoles and small chain fatty acids. These small chain fatty acids and indoles, they are directly have an impact to react with the brain, and they may alter the brain peptide release. So apart from that, these metabolites will also helping the digestive tract for the pancreatic reaction, for the normal digestive process, and for the circulation and for the absorption. So they will release the gut peptide, and that is also playing the role in the brain, and that will also having the impact on the feeding behavior or altered food intake of the host animal, particularly the fish, and it will lead to a altered metabolism. So this gut microbiota is playing a major role in any kind of the health of the fish. So ocean acidification, as I said, it is the seawater pH, the condition from 8.2, the pH is getting decreased. Also have an impact on the organ and internal environmental pH of many fishes. If the local environment pH is changing, definitely the internal environmental pH is also having an impact on the local environmental pH, and it have to react for that. So the fish gut microbiota will be directly altered with the biotic and abiotic factor like lifestyle, genotype, and physiology of the fish, as well as the abiotic factor like immune system, community members, and environment of the fish will alter the fish gut microbiome, and that will reflect in the changes in the phenotype. Okay, like the phenotypes means growth, healthiness, performance, even the reproductive performance, energy uptake, feeding behavior, and moving and escaping from the prey or predator. So this seawater pH is directly having an effect on the gut microbiome, and definitely it will have a negative or a positive impact on the particular population, microbial population of the gut. So what we did, now we know what is gut microbiome also, and what is the role of gut microbiome in the healthy fish environment. So what we want to do here is the experiment. We just want to use metagenomic analysis, and we want to mimic a ocean acidification environment, and we introduce some marine fishes in that environment, and we just study the gut microbiome population with the help of metagenomic analysis. So to know these three questions, will change in the seawater pH have an impact on fish intestinal microbiome? Will the fish intestinal microbiome play a role on the environmental information process? Because the pH is an environmental clue. Which will change in the seawater pH have negative impact on microbial signature. So we want to know whether there will be any impact on the intestinal microbiome, and we want to know whether environmental information process is getting changed, or there is any kind of negative microbial signature in the gut microbiome. These are all the questions we want to analyze through this analysis. So what we did is we just took a marine clownfish, and we just procured it from the ornamental fish traders in Chennai, and we maintained the fish in our laboratory with the proper aquarium setup, and we fed the fish with the proper diet like oyster, crab meat 3 times per day, and we almost keep it in the safe environment with a temperature of 28 plus or minus 1 degree Celsius, with a salinity of 34 plus or minus 1 degree Celsius, and with the pH of 8.1 and the dissolved oxygen 6.5. We have maintained the marine fish. And the light intensity was 14 hours light and 10 hours dark. And we did the seawater exchange for 50% weekly. Thus, we have maintained the fish in a controlled manner for 30 days before the experiment. In the experiment, what we did is we have maintained the pH in a different level with the help of a CO2 bubbling and setup. And the salinity is almost the same, 33.5 maintained in all the 3 groups. We got 3 groups, Each group, for the first group, which is the control one, which is 8.1 as a pH, and the experimental is 7.8 and 7.6. We have reduced the pH to 7.8 and 7.6 with bubbling CO2, carbon dioxide in the water. This carbon dioxide will definitely reduce the pH, and that is maintained throughout the uh, experimental period. But the salinity is almost constant, that is 3.5, and the temperature is 28.3. We have maintained that. The saturation also varies. The PSC carbon dioxide saturation varies. For the that is almost 580 and 1170 and 1941 as a partial CO2 was there in the control as well as in the experimental condition, and we almost exposed the animal for 15 days. The animal which we used is Amphibian tetula. So what we did is we collect the gut sample, the DNA, the gut was taken from the fish after the 50 days of exposure. The gut is the sample. We have stored the sample before DNA extraction, and we gone for DNA extraction with the commercially available DNA mini kit, and we quantified with the nanodrop to know the purity level, and we got a good purity of DNA. Then we gone for 16S rRNA gene amplification. Even for this amplification, we used a kappa hyphae hot start ready mix kit, and we use the commercial bacterial amplicons to for this particular amplification. After that, we gone for 16S gene library construction, quantification, and sequencing. During we use the commercial adapter dimer, and we do the purification process even after the library preparation also, and we done did the sequencing with the Illumina MySeq system, and later on that we got some kind of raw data that is taken to a bioinformatics sequence processing, where the two short alumni reads will be assembled according to the overlap, and FASTA-Q files will be processed to generate individual FASTA and QL files, which will be analyzed by standard methods. The sequence containing ambiguous bases and any longer than 480 base pairs will be dislodged, and they will and those with the maximum homopolymer length of 6 base pair will be allowed for the further process. All identical sequence will be merged into a single sequence. Sequence will be aligned according to the customized reference database. The competence of the index and the adapter will be checked and removed all the index and the adapter sequence. Noise will be removed using pre-clustered tools. We used for this particular purpose, we used a Mathur package as well as ribosomal database project that online tools. And we even detect the chimera, and we have trimmed the chimera also. And the species richness that and all will be narrated in the subsequent slides. On this particular data, the metagenomic data is being reposited in the NCB database, and you can get the entire sequence data in this biosamples. This is the identifier. You can get this, and there's a SRA, the sequential read archive accession number is here. So this is the rare fraction curve. Almost the rare fraction curves at a cutoff level of 3% were constructed at a 95% sequence similarity cutoff value. So this shows we got the OTU up to species level because of 97% of similarity cutoff we made in the rare fraction. And these are all the groups here. So see. One is pH 8.1 as a group, and pH 7.5 and 7.4, and normal seawater microbiome also we have taken in the water. Almost we got the sequence of 37,165, 30,302, and 36,411, and 36,455. Almost we got 97% of good coverage. And this is the community richness as well as the community diversity which we have done in this particular sequence. This is the alpha diversity as well as beta diversity in this population. And this is the overall OTU abundance in class level. So we got a variety of classes of bacteria. Even up to family level, we got a different kind of family. As I said, we used Mathur as well as RDP project, ribosomal database project URL. So these are all the URL. Even if you want to use it, you can use it. This is the at the different level. You can see a genus level, order level, and phylum level. There is a great level of changes in the microbial diversity. In the control, I'll see how it will look like and how it will look like in the normal pH and when the pH is altered. So this is the heat map, which is for a phylum as well as for bacterial genus. Even there is a great variation in the diversity of the gut microbiome. So these are all the unique OTU we have observed on this particular analysis. And these are all the various bacterial species we have identified. Though all the data may look unique, and you can say that yes, he is narrating everything, there will be creature biodiversity. But the main thing what we found out from this biodiversity analysis is the Vibrio population. So almost we have different kind of population from bacillus, from all different kind of phyla is there for all control pH 7.4, 7.8, and 8.1. But the Vibrio population, which is giving a kind of a unique microbial signature. So particularly we got Vibrio alginolyticus, Vibrio harvey, medrasicus, parahemolyticus, ponticus, and a clone called LC9. So in a normal pH, there won't be any large O2 of Vibrio. Even in 7.8, it is slightly increasing. But whereas in the case of 7.4, when there is a decrease in the pH, the OTU abundance of the Vibrio is getting increased. In the control, there is no Vibrio population. This increase may lead to a vibriosis in fishes. This vibriosis may lead to any kind of ulceration in the fishes. So if there is an increase in the vibriosis population, if they are pathogenic, if they converted to a pathogenic, definitely there will be a ulceration in the organs, internal as well as external organ. That may be the indicator for this ocean acidification effect in the particular fishes. So as I said, will change in the seawater pH have negative impact on the microbial signature? Yes, there is a negative impact. This Vibrio population is getting increased. So, but particularly this kind of analysis have to be again studied, and we want to confirm that whether it is really leading to a ulceration if this particular condition, this ocean acidification condition is persisted for a long duration. Actually, we have stopped the experiment in 15 days. It may lead to some kind of ulceration if we kept the experiment for more days, for 90 days or more than that. So such kind of studies we want to do in future also. We also done a pie crust analysis with the help of NIOT. We have done the pie crust analysis. I hope some of you know very well that even we can do Keck pathway analysis in the metagenomic sequence also. This pie crust particular online tool will help us this phylogenetic investigation of communities by reconstruction of unobserved states. So this will give the functional properties of the microbial genes. So for this also we did for all the 4 groups for control 8.1, 7.8, and 7.4. And we observed that The pH, actually pH may lead to some kind of osmotic regulation in the internal organ that may lead to a signal transduction thing, and the membrane transportation will be more affected. Because of this, maybe the fish is with the low energy, the fish cannot tolerate such kind of metabolism. If there is a high amount of the low amount of pH and high amount of hydrogen ion concentration, that may lead to some kind of osmotic imbalance that may reflect in the membrane transportation process here. So, will the fish intestine microbiome play a role on the environmental information processing? That is our question. Yes, we got the answer. When carbon dioxide is absorbed by seawater, a series of reactions release more concentration of hydrogen ion and affect membrane transportation process in the intestine. That is also be visible here in this Keck pathway analysis. So all the question is answered by this particular experiment. Yes, ocean acidification alters the fish intestinal microbiome signature. I saw all kind of colorful maps that shows there is a large amount of variation. And ocean acidification alter the fish intestinal microbiome signature and reflect in membrane transportation process. Even I show the example of Keck pathway changes, particularly in membrane transporters. Most of the pathway is working on those area. And ocean acidification alter the fish intestinal microbiome signature and increase the pathogenic. Yes, that's what I said. There is a microbial signature changes is there, but it is increasing the vibrio population. That is not good healthy sign for this ocean acidification environment. What I'm saying means there should not be any kind of change in the pH. The pH should be maintained at 8. If the pH should be maintained at 8, means there should be less carbon dioxide in the atmosphere. That is the take-home message what we want to confirm from this experiment. So to study the nature, we have to love the nature and stay close to the nature, and the nature will never fail us. So if we stop this carbon dioxide emission, means even with this experiment, even in the intestinal level microbiome population is also getting impacted indirectly with the because of this carbon dioxide emission. So we all have to work for the less emission of carbon even in our future thing. Thank you. Is there any question in the experimental process or in the PPT?
Hello, good evening, sir.
Yes, sir.
Sir, I am Dr. Milton from University of Madras.
Yes, sir, please, sir.
So, very fantastic talk on this fish microbiome, and I have some series of questions if you bother.
Yes, sir, please, sir.
Don't bother. So, what is the adaptability of these fish species that you have taken, as well as in general we can speak?
Yes, sir.
Like that of the thermoregulation process, is there is any adaptability to that of the pH that could be changed in that particular environment? What do you feel about that?
Sir, definitely for any kind of environmental changes, definitely these fish will get the acclimatization first, then they will get adapted to the environment.
Okay.
But here, as I said, generally people normally people will do acidification work with the help of acid based induction.
Yes.
So what we did in our experiment is we almost bubble the carbon dioxide in the environment. So we just want to know whether this kind of carbon dioxide bubbling and the change in the pH, whether the fish is getting sustained in that particular environment.
Okay.
But we did the experiment only for the 15 days. But there is no changes in the external phenotypic, or there is no death in the fish.
Okay.
But there is a change in the intestinal microbiome that is reflected but I cannot say that the fish adapted to the change of the pH. We have to do some more studies also here. But as there is no lethality of the particular population, I said maybe these kind of fishes are trying to get acclimatized with this kind of environment.
Yes.
But during this acclimatization process, that will lead to the adaptation of this environment. There will be internal changes.
Yes.
That internal changes are reflected in the microbiome, particularly in the gut microbiome. That's what we want to hypothesize in this experiment.
Okay, okay, sure, sir. Thank you very much.
Thank you,
And you have taken that one experimental fish for your study.
Yes.
So, what about that the gut microbiome that could be available in the marine fishes? Is it having that same type of these microbiome, or it is different with each and every type of species?
Sir, individually, the microbiome will change to every species, sir. There will be every species, but when we are having a genus level, the abundance of the OTU will have some kind of percentage similarity. We cannot say that there will be a species level similarity. The genus level abundance of percentage similarity will be there, but there will be some kind of difference in the species level. The percentage of the significant level of the fold increase in the particular genus level, that will be have to be calculated here. But every individual, even we all have some kind of unique signature of microbiome. So it varies, depends upon the individual species. But it is unique for all kind of genus. With some kind of a percentage of OTU. Yeah.
Okay. So what about that skin microbiome? So do you have any research on that particular area? Apart from that of the gut microbiome, it has any impact on that?
Actually, we have not done anything on that, sir. Actually, I came across one reference, but what they have did is they have just isolate the slime from the fish skin, and they worked on the gut microbiome. Sorry, skin microbiome. Particularly the microbiome which is dwelling in the slime of the skin. But the experiment on the end of the experiment, they said nothing happened to that microbiome in the skin. Like that, they have concluded that particular paper. I just forget the name of the author on that.
Okay, no problem. So the next one is what could be the impact of our gut microbiome when we have eating such a kind of fishes that grown on that particular acidified environment? Is there any impact on our human gut microbiome?
So definitely, sir. Not only that fish. If we are if you are taking any kind of
concerned about that thing, when we are eating that marine fishes.
Sir, not only marine fishes, sir. If you are taking any kind of food which is not in the proper form, if it is getting decayed or anything in the abnormal form, if you are taking that, that will definitely have an impact on the gut microbiome of human. And definitely, this Vibrio population is more in that particular fish.
So leads to that of ulceration.
That also have a great impact on us. But the only thing is, I have not worked on any kind of seafood. I mean, food fish. This experiment was carried out in the normal.
You have not done any fishes that could be in the natural environment. You have not done anything.
So actually, this is the preliminary work we actually we did that, and we are planning to work more on this aspect.
Okay. Thank you, sir. Thank you very much for your nice information and the highly informative one.
Thank you, sir. Thanks
a lot.
Good evening, sir. I have a few questions to ask.
Yes.
Very commendable work, very extensive work. Happy to see such a good work being presented. To add on to the question what Dr. Milton has asked, he asked about the gut microbiome of clownfish, what is found in the natural environment, whether you have done a comparison or whether you have checked that also when you have collected this. And also, have you checked for what is present in the seawater from where you have collected this? That is my first question.
Actually, I show 4 groups. One is the seawater microbiome. Okay, and when we collected all the fish, actually we cannot do immediately, but we maintain all the fishes in the same environment. That's what for the 30 days of period I maintain in the normal pH. So what I thought that may be the control for that. So for all the fishes, so we give a normal food. Generally, clownfish will eat clam and all. So we provide the same food, and we maintain the normal temperature, normal pH, and normal aeration. Even the light intensity also, we maintain the same as in the sea for 30 days period. And the normal thing is taken as a control here in the 8.1 pH. So that will be the answer for your thing. If we have done anything directly in the environment. When we took the sample, I have not done anything on that, but I acclimatized entire population. Then I worked on that. So I just guessed. I got a conclusion that this 8.1 is the normal gut microbiome when we done the experiment.
But sir, don't you think that you need to have one more as a reference from on the spot collection and to compare with both seawater as well as from the
Yeah,
fish?
definitely what you're saying, if we have a database or a baseline data on that, if we are going for such kind of experiment, it will be very nice. But due to that economical constraint, we have stopped this experiment in this level.
Sir, and why you have kept the fish number as 6? Don't you think it's a less number? Because when you're doing such kind of experiments with varying the environmental parameters, don't you think the chances of mortality could be more? Because
As I already said that actually this is my idea. What I want to do it in a bigger way. Actually, even I want to create a setup for the social certification. So we are working on that. For the preliminary data, this much population is because I have to get the ethical clearance also from the committee. They said, actually, I asked for a 12 number, but they said, no, you just go for 6 number. As per the committee says, we just go for 6 number. As you said, it is very less number for a preliminary data for this particular experiment. It's a preliminary experiment. We cannot get any kind of a strong conclusion, but for this. But from this way, we can work on any other new hypothesis. For that, this kind of a small population, sample population is, I think it is okay.
sir, and whether they were triplicates for each pH, each tank, I mean,
No,
like
no,
8.1.
we have not done any because this kind of setup to maintain this setup also, the cylinder, CO2 cylinder, we have exhaust a lot. So as I again, and this is the answer for me. This is a preliminary work, so we plan to do in a very micro level. That's what we limited everything. But when we took the sample in a triplicate manner, the gut microbial samples. When we gone for the genomic analysis, we gone for triplicates.
Sir, and for keeping it for the conditioning purpose, you have kept for 30 days. Yes. But for experimental setup, you have kept for 15 days only. So yes. Why is that? Do you think that 15 days is enough for the change to be exhibited?
No, actually, I thought to go do this experiment for 100 days. But as I said, no, when I started bubbling the CO2, there will be excessive need for carbon dioxide bubbling to get the actual pH condition. So using more amount of seawater, again, I'm doing the same thing. I'm just bubbling all CO2 in the atmospheric only. And as per ethical committee, they asked me to go with such kind of a quantity of CO2 alone. So we thought to stop the experiment on the 15 days. But if I did the experiment for 100 days, maybe I got the answer for what Dr. Milton have asked me, whether this fish is getting acclimatized or adapted for that. As there may be another possibility, we can work on some kind of transcriptomic aspect of organelles also. But due to some kind of limitation, we stopped the experiment in 15 days.
And sir, you have said that weekly once you have changed 50% of seawater is being changed.
Yes.
So there is my question is that is it from the same area you were able to do the seawater, or when you're bringing it from a near shore or something?
For this, I request all the participants as well as allied to come to our Center for Ocean Research. We have a really fantastic facility with us. Almost 50,000 of seawater samples with us, and we have a large amount of sea recirculatory facility and all. and even we have one more facility like that, and any kind of organism we can rear in our facility. So that recirculation process is easily available. I'm able to maintain the fishes, and I can done the experiment in that manner. That's all.
Sir, actually, I asked this question because most of the cases in our labs and all, when we are doing, we have to collect because of the logistical issues. We cannot go away.
That's what. Yeah.
Collect the water. But
So,
when you are dealing with means microbial world is so sensitive that it will change.
yeah,
Even
definitely.
the waves, tides, every time it is changing. So when we are bringing it and keeping it in a stagnated condition, still the microbes will be going for a change. No, that is why I just thought like I asked.
That's what I'm said. So that definitely there will be a change when the fish will be taken from the wild to the lab.
Yeah.
But in the 30 days of acclimatization period, almost the feed what we are giving, and the animal is only living in the environment where the water is given by us only. So sure, the animal will be set to the a microbe present in the water as well as to the host microbiome, that there will be equilibrium that will be developed inside the host of the fish. That will be almost equal to all the animals, experimental as well as the control animal, on the end of the 30 days. So that's what the acclimatization period is, 30 days, and the experimental period is very less.
Okay, thank you, sir.
Thanks a lot. Thanks a lot.
So there is a question in the chat box, sir.
Yeah.
Sir, actually the sea fish intestine or viscera is removed before cooking. Then
Yeah.
how it affect our gut microbiome and when consumed cooked fish food?
See, if it is properly removed, definitely there won't be any problem. If it is improperly removed or any kind of improper cooking, normally all kind of the seafood based, this is all because of the non-proper preparation and semi-cooked food only. So if we done everything normally, it is well and good. And if there may be, this is the thing. That's what I want to say. All whenever we are going for a seafood-based disease and all, they use generally they used to tell that Vibrio population are more pathogenic and providing more kind of disease. That means everyone is cleaning the viscera and all, and they are doing proper cooking, and then only they are eating the seafood. But in some case, it may happen to cause some kind of pathogenesis. That time, this kind of overpopulation of Vibrio should not be there. That is the answer. But as he asked, definitely by removing this and with the proper cooking, any kind of bacteria can be destroyed.
Okay, sir. Thank you, sir.
Sir, have you found any mortality in any of the experimental setups?
No, we don't have any mortality. That's what I answered to Dr. Milton. There is no mortality in that fish. As there is no mortality, I can say that the fish got adapted. That is that kind of experiment I have not done. That's what I said. As he asked me whether this fish is adapted to this particular environment, but there is no mortality and there is no changes in the phenotype of the behavior of the fish. But there is a when we done some this kind of intestinal gut microbiome analysis, we saw this changes. But there is no specific changes in the fish during this experiment.
Sir, and also one more question, like when we are collecting from the wild, how can we be sure that the fish all belong to the same period of age or something like that, you know?
No, actually, we request the fishermen to bring almost, as I mentioned there, the length of the fish and the weight of the fish, almost what we request they used to bring to our lab. So generally, whenever we are asking to the fishermen, we need this fishes and this length means definitely they will give. Actually, in this experiment, we ask for 5 centimeter length of fishes. So almost every fish will be 5 to 5 or plus or minus 0.5 centimeter length will be there. So by that I can group the fish in the same length. That's the only thing I can do. If we, as you said, if we want to know what is the community level thing and even gender wise and all, I have not grouped the fish. Just taken the fish in the length, as I said. If it is in 5 centimeter length, I thought it is all the fishes will be the same age, so that I just took the fishes in the same manner.
So one more question. It may seem funny, but after the experiment, did you release it back to the?
Actually, I should not say this answer here. The biggest problem is we sacrifice the animal to get the gut sample. So for that purpose only, this ethical committee people are not providing more animal to do the experiment. So if we are keeping 100 animal, if we are getting the 100 animal, means the entire 100 population will die. So that the ethical committee people, they stop the sample size and they are reducing the sample size.
Okay.
So,
but sir, do you think it is still valid? No, like even if we are reducing the number of samples, when it comes to the publication point of view, it is still acceptable, no? Because we have a valid reason because of the ethical issue. We have reduced the sample number.
That's what I said. This is a preliminary experiment. We cannot, by this, I cannot say that definitely ocean acidification is having a great effect on Vibrio. I cannot give that kind of hypothesis. I cannot give. But my experimental is one of the baseline data for such kind of experiment. If you people are doing that experiment in a large sample with a large amount of experimental setup, definitely this will be a reference point. That is the only conclusion I can give here.
Okay. Thank you, sir.
Thank you. Thanks a lot.
So one more question, sir. If all the organisms adapted to live with the lower pH, why we worried about the ocean acidification? I think that you have like
all the animals are adapted to lower pH. Definitely, we all are also adapted to any kind of environmental change. But whatever we are getting adapted, even the fish is getting adapted, there will be a phenotypical or a genotypical or internal morphological or molecular level changes will happen in the environment because of any kind of environmental changes. So we have to worry about it. See, if the Vibrio population is increasing and that particular Vibrio is causing ulceration in this particular clone fish, for example, if it is exposed for a long period, definitely the entire population will lead to death. If this happen in an environment, if the population is getting decline, means the population, the dependent population of that particular fish may also get decline, and that may lead to a change in the food chain. That's what we want to see. So here he is asking why we have to so worried about, means definitely we have to worry about it. Though it is not visible to our eyes that the fishes are not dying, means it is not that the fishes are happy. So there may be some problem in that. Even the pathogenicity is increasing in the lower pH. That's what I want to conclude here. Hope I have answered Dr. Rajendran's question.
Yes, sir. He had
Thank you.
given.
Thank you. Thank you. Thank you.
Thank you, sir. Thank you so much for the detailed presentation and detailed explanation in the question answer session also.
Thank you. Thanks a lot. And tomorrow is the final day. There will be two sessions, and one is about the or statistical analysis, and that will be given by our HOD of Department of Biotechnology, Dr. Ramesh sir. And another one is why we need ocean and sustainability. That will be taken by Dr. Sheshatri sir, who is the TBI coordinator. Both the sessions will be very informative and interesting. So please join tomorrow, and I request everyone to give feedback on the final session. Thank you, everyone. Thanks for patiently listening to my talk as well as Mr. Kishore's talk. Thank you. See you tomorrow. Thank you.
Thank you, sir.